We're going to take a look at the cultivation of viruses and viral taxonomy. Can viruses be cultured like bacteria? Yes, they can, but only if you grow them inside cells. We don't do viral cultures on patients very often because it takes a couple weeks to get the results back from the lab. And we usually want to have answers for our patient and get a diagnosis pretty quickly. So there's some other ways that we can diagnose a viral infection in our patient. So one way we can do that is we can see if a patient has antibodies against a particular virus. So for example, if we want to see if a patient has HIV, we can look for antibodies against the HIV virus. And if they're present, then the patient is HIV positive. We can also look for viral antigens. So for example, spike proteins. And if those are present, then the patient has maybe, for example, COVID-19. We can also look for the viral nucleic acid. So for example, a PCR test for COVID-19 would look for the viral nucleic acid for the SARS-CoV-2 virus. And if that's present, then you would be positive for COVID-19. So here's a viral culture. This one is for bacteriophages. So we have some bacteria growing in our petri dish here. And we've put the bacteriophages into the culture media, and they've infected the bacterial cells and killed some of the bacteria here. And you can see those clear areas where the bacteria have been killed. The clear areas are called plaques. And then we count the plaque forming units, and that will give us an idea of how many bacteriophages or viruses that we started with originally. We can also culture animal cells in cell cultures, and we can count the number of these viruses by counting the number of plaques, these clear areas where the animal cells have been killed by the virus. Or we can also count the number of cytopathic effects as well. And we'll look at those in just a minute. And this is just showing that some of the viruses have to be grown in not only cells, but sometimes they'll grow them in chicken eggs. So like the flu virus. When they grow the flu virus to make the flu vaccines, they will grow them in chicken eggs, because the virus needs to grow in cells in order to produce more virus, and chickens are one of the species that are infected with the flu, and so they will use chicken eggs to grow the flu virus. All right, so some of the animal viruses actually don't kill the cells right away, but they can cause some damage to the cell or change the cell's appearance. So we call these cytopathic effects. So then we can look for those when we detect the virus. And you don't need to know the other parts on this slide. I won't ask you those. So here's an example of some cytopathic effects that are caused by a virus. So over on the left, we have some normal cervical cells, but over on the right, we have some cervical cells that have been infected with herpes simplex virus type 2, or the virus that causes genital herpes. So this herpes virus will cause the cervical cells to fuse together and form a giant cell with multiple nuclei, because the cells have all fused together. So this giant fused cell with the multiple nuclei is called a syncytium. So syn means together, and cyte is talking about the cell. So when you see a syncytium like this, that indicates there's probably a viral infection going on, because we've got this cytopathic effect there. So viral classification is actually pretty hard because we don't have a good evolutionary history of viruses. There's no fossils of them. And we really actually don't even know how they originated. So where did they come from? We're not really sure. There are a couple of groups of scientists that have come up with some classification systems. And to date, there's been over 9,000 viruses that have been studied and have been classified into 59 different orders, 189 different families, and over 2,000 genera or genuses. And we know that there are a lot more virus species out there that are still waiting to be discovered. And as far as taxonomic classification of viruses, this is very similar to what you've already studied before. So there's different orders and families. We have genus, and then a viral species. That's just defined as a group of viruses that share the same genetic information and the same host. A lot of times we use descriptive common names for species. And so, like, for example, we have the measles virus or the mumps virus. And then a lot of times, if we have like a subspecies, they oftentimes give them a number. So with the HIV virus, there's HIV-1 and HIV-2. So that would be an example there. And this is just an example classification of the mumps and measles viruses. You can just look at these. You don't need to memorize these. But the mumps and measles viruses are evolutionarily related to each other. So they are actually in the same subfamily, but they're in separate genuses and obviously separate species. So as far as classifying viruses, there's a number of ways that we can classify them. So we could classify them based on what type of nucleic acid that they're using. So remember, there's 4 choices that a virus could choose from. So some viruses will use double-stranded DNA as their genetic material. Other viruses use single-stranded DNA. And some viruses use double-stranded RNA, and then others use single-stranded RNA as their genetic material. So they will choose one of those as their genetic material, and we could classify them based on that. We could also classify them based on whether they have an envelope or not. We could also classify them based on the shape of their capsid. So is it helical? Or maybe it's an icosahedral capsid, or maybe it's complex. We could also classify them based on the size of the virus. So that would be another way we could do that. But the classification scheme that we're going to look at is called the Baltimore system. A scientist named David Baltimore came up with this idea of how to classify viruses. And what he did is he organized viruses into 7 different groups based on the type of nucleic acid they were using as their genetic material, and then also how they replicated. So this is the Baltimore classification system. These are the 7 groups that he came up with, and we're going to go through these 7 groups. You do not need to memorize these 7 groups and which nucleic acid they're using, and I'll show you what you need to know about these. But what's interesting is that groups 6 and 7 here, notice that there's a RT on each of these, and that just means that both groups 6 and 7, they're able to do reverse transcription. And we'll talk about that, but they're able to do transcription backwards. So they're able to take the messenger RNA and from that make DNA, which our cells cannot do. So for example, in group 6, we have the HIV virus. And in group 7, we've got hepatitis B virus. right, and then just kind of in general, taking a look at our animal viruses. If an animal virus is using DNA as their genetic material, they will replicate the viral DNA in the host cell nucleus because they're going to be using the host cell's enzymes to make more of the viral DNA. And that's where the enzymes to replicate the DNA will be located, is in the nucleus. And then they're going to be making their capsid proteins out in the cytoplasm because that's where the ribosomes are. So that makes sense. And then animal viruses that are using RNA as their genetic material, they do things a bit different. And they're going to be using enzymes that our cells don't have. And so they're going to be bringing these enzymes with them or making them right when they get into the cell. So what they're going to be doing is they don't even need to go into the nucleus because they're not going to be making DNA, and so everything they need they can just do right in the cytoplasm. So they don't even go into the nucleus.